Amanda Prieto, and I'm the founder of Amanda Oceano. Amanda Oceano is an organization with the mission to inspire love for the ocean and its ecosystem. We are here today as part of a series called Conservation Conversation, where we interview people, organizations, and more of the different ways they are raising awareness for the ocean. Today, we're here with Chelsea and Evan, a couple who found their place here in Puerto Rico and founded Isla Mar Research Expeditions. Currently, Isla Mar is working on a project investigating the use of eDNA to locate fish spawning aggregations, as well as several other projects, including identification of critical habitat use by juvenile Nassau Grouper, which we will be speaking about soon, evaluating the status of parrotfish in the Caribbean for the FAO, monitoring nurse shark aggregations around the island, and much more. Right now, I'm going to give the space to my friend Amos to introduce himself and so that he can say the rules of today.
Hello guys, I am Amos Santiago. I am part of Amanda Oceano. I'm secretary and project manager as well. I'm going to tell you a few rules, at least for the ones who are meeting us through Zoom. Please maintain your mics and cameras off at all times. Please also limit the topics of your questions off of politics and religion. The interview will be approximately 1 hour. Questions or comments can be typed through the chat while the interview is happening. I will be also translating if you want to ask those questions in Spanish. And you can also send those messages through Facebook. The questions will also be answered at the end of the interview. I'll put a link. Did you already put I'll put the rules in Spanish in the chat just in case you want to read them.
Yeah, I put them in English and you can put them in Spanish.
Okay, perfect. So let's start. Hi guys. I'll be asking a few questions. So can you tell us what is Isla Mar and what brought you to found this company?
Sure. So first, we just want to say thanks for having us tonight. We're excited to be able to talk with you guys and with your organization's members and tell you a little bit more about like what we do. So Isla Mar is sort of like our vision with a mission to train the next generation of scientists in essentially field biology as it relates to the marine systems. And for those watching that are graduate students, I know that Amos and Amanda and I were kind of talking about this just a little bit before we got started, but we were telling them about how when we founded Isla Mar, we were both still graduate students. And our company first started as a way for us to teach in the field. So we both have had interest in being able to teach students, but kind of in like a more non-traditional setting. So not necessarily in the classroom. And we had found our way to Puerto Rico by taking a field course when we were undergraduates at Florida Institute of Technology and kind of fell in love with the island there. And we got to spend 2 weeks actually at Isla Magellas, part of UPR Mayaguez. And we got to just experience what it was like to be able to work in the marine systems that are so close here to the island. And we fell in love with that like accessibility and just the ease that there was there to jump right into projects. And so that's kind of what brought us to Puerto Rico. But then over the years, Florida Tech continued to bring students down here, and we would essentially help guide those field courses, because then now we were the ones that were living here and we're kind of familiar with the area and could help facilitate basically that whole course. And that's kind of how we got started, is really just in the leading and the organizing of the field courses. And so we actually started doing that in 2012, but we officially became Isla Mar in 2015. And from there, it turned into a little bit more of a, besides just the education and leading the field courses, we also started writing our own grants. And so it became a little bit of a consultancy in a way, because we wanted to be able to like use our expertise and our skills that we had developed through graduate school, but to still work on projects that interested us. And so we started writing for federal grants and for state level grants and for projects that interested us, which were typically in things like fisheries management type focus for most of it. And that's essentially what has led us to where we are now, where we've been here on the island now for 9 years. And yeah, so Isla Mar is we kind of will spend the summer times doing our field courses, but then the rest of the year is when we do the majority of our other research. So it's a little bit of both.
Yeah, it's kind of like an interface between academia and like in the private sector, right? So
it is.
Mm-hmm.
Yeah, and that's I would say really it's kind of like a way to look at like the new independent scientists, I think, because consulting has been around for a while. Like that's not new really to the marine sector. Like you have people that are independent scientists that just consult. But what we've tried to do is kind of combine that as well as like the teaching aspect of it, which yes, is traditional in like an academic sense, but neither of us are professors at a university, but we have that interest in being able to offer something that sometimes students can't get in a university setting, especially if they don't have access to the marine environment. So that's kind of where like leading the field courses, we found sort of that opportunity to be able to give students a more hands-on and like a hands-on approach in the field that a lot of times is lacking in the classroom setting.
Yeah, definitely, I would agree to that. Now that you talked about consulting and stuff, you guys were recently part of a global project where you collaborate with HJR Reefscaping to document sharks around Puerto Rico, right? Can you tell us a bit more about that experience?
Yeah, that was a really cool and unique experience that we're very fortunate to be a part of. We were involved in like the very early stages of that project. Because as you know, Hurricane Maria came through and kind of rearranged everybody's day-to-day lives and what was possible and not possible. But we were able to take part in the project from early on. It was a really big project, multi-collaboration, multi-agency, multi-country project. If you've seen the paper that just came out, you'll get an idea of actually how big and the scope of that project. But we were able to participate here in the Puerto Rico portion of it, and it was a unique experience for a lot of us involved with that project. And with HJR Reefscaping, it was our first time working with BRUVs, which was the primary mode of data collection for that project. And they are basically baited remote underwater video systems. So you have a tripod with a GoPro camera with a long stick with a bait bag on the end of it. And you drop those off, you deploy a string of those, say 5 or 6 at a time, leave them for an hour and a half to 2 hours, and then recover them. The idea being that you have a video capturing what's going on in front of the camera or with the bait bag, and then the bait should attract sharks. So it was a very cool way and really like into like a new and initiative way of actually surveying shark populations without being in the water, because a lot of times, believe it or not, sharks tend to shy away from divers. Divers underwater are really noisy, but then also the attraction, having chum or bait really helps with that as well, because there's not that many divers that want to sit there and swim around with a bag of dead fish. So but yeah, it was really cool. The project basically encompassed the whole island, Mona, the west coast, the south coast, and some areas on the north coast as well. And it took, I think, a year and a half or so was the entire data collection series of that. We weren't involved with all of that, but we were in the beginning, and it was added to the global database for global fin print. And they got a really good data set out of that, and they were able to show some of the conclusions of that paper were on many of the reefs around the world that sharks are functionally extinct. They're not there. They're not in high enough populations, to really make a significant ecological effect on the ecosystem anymore.
Wow. So it's no longer like some species have just like disappeared from.
Or not necessarily disappeared, but their numbers, their abundances or densities are so low that they're not as encountered as frequently as they should be.
Wow. And in the case for Puerto Rico, what results?
That was, very, very similar to that. Yep.
Okay. We have a question in the chat. What was the max depth of the baited shark survey, and what sharks were you surveying?
So the maximum depth for that project was, I believe it was 40 meters. So that's about 130 feet were the deployments. The majority of them, you know, so a lot of them are shallow or much shallower than that. A lot of them were done just via snorkeling and free diving. You set the tripod, you drop the tripod and set it up, snorkeling and free diving. But on some of the deeper ones, we did jump in the water with scuba. Because you're carrying a heavy object, a weighted object down with you. And the different types of sharks that we're surveying was basically anything that would be attracted. It was basically just seeing what was there and what was in the ecosystems that were being surveyed. So really any shark that would come up to the camera was fair game. I'm not sure the total number of species that we're seeing in Puerto Rico, but you know,
there were nurse sharks,
reef sharks, you know.
Yeah. Hammerheads, I think, were in some of the videos.
Yep. Yeah, so, that's right. Yep.
Yeah.
But then also too, it gives us some baseline as well, because you know, in some of those areas, or even with that methodology, it just doesn't attract sharks, but it also attracts some other piscivorous or other fish that eat fish that would be attracted to it as well. So you know, it gives, you know, those types of projects, you know, they might focus on, you know, one specific group of species, but really you can gain a lot of information from different types of projects like that.
Nice. What specific sites were chosen in Puerto Rico?
So Rincon, Aguadilla, some areas off the west coast, like Bajo Rico, Mona, La Paguera, believe Salinas.
That might have been as far.
And I'm trying to think, I know there were more areas, Ponce, but those are the ones that come to my head right off the bat.
Awesome. Thanks for sharing that.
Mm-hmm.
The next question we have, what is the current status of the species lionfish here in Puerto Rico, and how is it ecologically relevant?
Yeah, so I guess before we talk about like the status of lionfish in Puerto Rico, I think it's important that I mentioned that our invasion here in Puerto Rico was never quite as excessive as it was in places like the Bahamas and like the Western Florida Shelf. So even though lionfish you can now find them everywhere within the Caribbean, we never had the same density as places like the Bahamas, and so we were never really seeing the same kinds of impacts that those sites were. So that was actually something I did for my PhD, was study the effectiveness of using small scale removals for controlling lionfish populations, and basically like what's the response, like when you're putting all this effort into removal, what's the response of the native fish, primarily like the prey species or the things that we're thinking that they're eating, and how are the other competing piscivores or other fish that are eating other fish, basically kind of along the same feeding strategy as lionfish, how are they responding? And essentially what I found is that at our kind of like low lionfish densities that we had on our reefs, and this study was done in Parguera, that we're really seeing no impact of lionfish to our prey communities and our piscivore, our native like predator type communities. So with that being said, I do know that it's getting more challenging for fishers to find lionfish, that there is some demand for lionfish in restaurants, and so they are asked to go out to try to meet this demand, and I know that they have had some trouble finding them, but there are sites that people, you know, there are some places habitat wise that they do still prefer. For instance, there was like an ROV, a remote operated vehicle study that took some video at about 70 meters off of Mayaguez, and just within the frame of the video, you couldn't even count the number of lionfish that were within that shot. So there's some places that are not going to be possible for us to really access in order to control them, and whatever that means. Some folks have suggested traps, which is great for areas like the western coast of Florida, where you have this really like expansive flat shelf system, but it's not really feasible for places like us or for other like smaller island nations that really don't have that kind of a system where you do find lionfish in great densities. But yeah, on some of our deeper reefs, they can be found, but they're not really accessible for us to harvest them. But in terms of our need to be concerned about it, I think that at least at the time when I was performing the study, it wasn't as alarming as it was for these other countries that I mentioned. But I mean, that's something that should be likely reassessed. But I think that the latest paper that's come out, though, speaking in terms of the Caribbean in general, has seen a general decrease in their populations, at least for most of the Southern Caribbean. And that study in particular was referring to Southern Caribbean as essentially like the Greater Antilles and South. So yeah, and then in terms of like what's the ecological importance of that, well, you could look at that in like a couple ways. I know that initially, and I know that there are still people kind of looking into like a market strategy for lionfish or trying to bring them into the fishery as a way to eat them. You know, that might work for some countries, but might not for others, like in places where you don't have such a great abundance of them, then that's maybe not serving the same role in the sense of trying to take pressure off of some of the other fish that we are fishing. I think that was what kind of the idea would be, but that might not work everywhere. And then in terms of like how they might be competing with some of our native species, really, I think that it's going to be more in terms of competing for space if they are, because lionfish really prefer crevices, caves, that kind of thing. Groupers also like those kinds of habitats as well. So if there's competition in that sense, it's probably going to be for space, not necessarily for food resources.
And do you know how the lionfish came here to the Caribbean, or there's just like theories?
Well, so some things, I mean, I'm not sure if there's been anything recent on this, but I believe one of the theories, which would make a lot of sense, is that they were able to spread throughout a lot of the Caribbean through ballast, which that seems more plausible. Like ballast water that's in ships that gets released when they come into port, or near the islands as they're coming in. So that could be like larvae, the eggs of the lionfish that are caught up in that ballast water that gets released and then they settle. They can be in the water column for like close to 30 days. So it's actually a pretty long time to be able to hang around. So they also can drift, especially with like neighboring islands, they can drift just with currents and stay in the water column until they hatch and settle. So, but, for those longer distances, I think that the current hypothesis is that it was probably ballast water.
Okay. And I have another question. Do the lionfish juveniles, or do they carry the venom also, or just like the adults?
They do. In terms of,
it still hurts.
Yeah, I mean, when you get, well, I mean, so their spines are like hypodermic needles, so that's gonna hurt regardless,
Okay. Okay.
But yeah, I mean, the juveniles still have the venom, but really, in terms of like getting envenomated by a lionfish, I would say it's more about like how far the needle really gets.
It's a surface area. If you look at the spines on the fish, they have a fleshy membrane that goes over the spine, and that really kind of encapsulates the venom around the spine. And so it's more of a surface area. However much spine you have going into your finger, the more venom you become envenomated or in contact with. And that really more or less affects, I'm sure, the severity of the injury from the envenomation.
And so I guess this would depend if it's like the frontal or the anal fin, or is this the same thing?
Going on the same like surface area aspect of it, the dorsal spines, the ones on the top of the body, those are the largest. So potentially those could, have the highest surface area to human contact. But just in terms of, for the same amount of venom, the same quantity of venom, I'm sure, I don't know, but I'm sure it all hurts just as bad.
Yeah, definitely. Have you been stung by like a lionfish?
Yes.
Yes, we both have. It's just
Damn.
you work with them enough, and it's inevitable.
It's inevitable. Yeah.
Yeah. People have like compared it to the scorpion fish, but they say the scorpion fish is like a million times worse.
Mm-hmm.
I wouldn't know. I haven't been stung by anything else.
Yeah,
I
haven't been stung by a scorpion fish. The way I usually try to describe it is it's like just a very intense burning and throbbing. So it's almost like you take the area, say you get stung in the finger. If you were to take your finger and put it on a hot frying pan and then smash it with a hammer for about 4 hours or so. Sometimes longer.
What? For real?
Yeah.
Damn, wow. So definitely the hot water is the best, you think, like.
I mean, it helps a little.
Well, the venom's a protein, so extreme temperatures denature the protein and help it break down, but it's not instantaneous relief by any means.
Okay, thanks for that. We have a question in the chat. Graciela says, any plans to develop gadgets that can harvest lionfish at depths beyond diving depths?
Yeah, I've seen a couple things come across. I know that there's a group that's been working on traps. They've primarily been deploying those in the Gulf of Mexico and like the Western Florida shelf. And they found some pretty good success with that. But like I was saying, like it kind of depends on if you have the right habitat really to drop those like in series. And I have seen, I don't know how far this has actually come, but I have seen like an ROV that has a little contraption on the front that I think you have to maneuver the ROV to get the lionfish sort of in between these two panels, and then it sends an electric shock between the two panels, and it's supposed to stun it. And then I think they have like a suction where they can vacuum it up. But I mean, that's honestly, I think the traps would have better success than than the ROV. But you know, I guess people are always coming up with unique ways. So I, you know, would like to see what we have in the future. But I think the traps have a lot of promise.
I have a question. How big of a problem is the lionfish in other places like Florida and those coasts?
Well, there have been several studies that have found that in pretty high lionfish densities are having an impact on the native fish there, in terms of like what we would consider prey items for the lionfish, but then also to for some of the native like predator species, so things that would compete with lionfish for both space and for food resources. So the Bahamas and Florida initially, and even now, I'm not sure what their status is currently, but they, yeah, we're dealing with pretty heavy densities of lionfish, and they were seeing some pretty extreme impacts from them. And that's kind of what sparked sort of like the rest of our inquiry throughout the rest of the Caribbean, because that's really what all of us then south of those two areas were expecting to see. And so we were kind of readying our arsenal for that in anticipation that we would see the same thing, but not all islands south of Florida and the Bahamas saw those same results.
Wow, intense. And like fishers, wait, we have like a question here. Are Nassau Grouper or other larger predators eating lionfish here in Puerto Rico? Any lionfish in the juvenile habitat are you surveying?
I've seen video of Nassau eating lionfish, but it was in the Caymans. Have you ever seen Nassau eating lionfish here?
No, I have not. Like there are some videos of sharks, you know, larger groupers, larger snappers like mutton snappers, eating lionfish. I have seen, you know, divers here feeding lionfish that they have killed to sharks and eels and things like that. But personally, like I've never seen or heard of lionfish. I mean, larger predators here naturally, meaning not being fed by divers, feeding on lionfish.
Right, and on that note, that's a very poor practice to teach a predator to eat a lionfish, because that basically just stays with that individual fish. That's not something that's passed on, because that that is something that evolutionarily those fish have to learn and pass on themselves. So just teaching one individual predator to eat a lionfish is just doing that. But not only that, but it's teaching also that predator to associate a diver with food, and that can become really dangerous.
Yeah,
then,
because
go ahead.
Yeah, like, keep on. What were we gonna say?
I was gonna say, and then yeah, the second part of that question was, are there any lionfish in the juvenile habitats that we're surveying? Yeah. Yes, like for the juvenile habitats of Nassau Grouper, we are seeing lionfish in those habitats.
Cool. So you wouldn't say that there's like a natural predator for the lionfish?
There's no natural predator for them in the Caribbean.
Right.
And it's not very well documented what their natural predators are in the Indo-Pacific, because they're not nearly in the same abundance in the Indo-Pacific as they are here. Like, you don't see them as commonly on the reefs there, like how you would in the Caribbean now, going around and seeing lionfish on the reef here. There's also many other species of lionfish over there versus what we have here too. So there's going to be more competition over there than here.
Yeah, and I guess like the species here like don't associate that particular fish with food, maybe.
Exactly. They didn't evolve with it. Exactly. So they don't recognize it.
Yeah. Oh, interesting.
Wow. So what predators like do they have in the Pacific? Would be like sharks or
it's not well documented. Yeah.
Oh,
I mean, it's gonna be like other fish that like other piscivorous fish. So bigger, probably bigger grouper and sharks. Yeah. Eels.
And I don't know if this like is a relevant question, but how do they get rid of the venom? Like, how do they not get affected by the venom when they eat lionfish?
Well, a lot of fish tend to consume their prey head first. So when they do that, the spines actually lay down. So for fish that do that, then they don't necessarily have to worry about that because they're not being envenomated by it. Because the lionfish meat itself is not poisonous or venomous. It's just the venom that's located in the spine, and so the only way to get that venom out is if you, if you, what's the word I'm looking for? Like if the spine is disrupted, so that the sheath that's covering the spine is removed, I guess, in a sense, and that's how the venom will penetrate with the spine itself. But yeah, I have seen eels. I've seen videos and photos of eels that must not have eaten the lionfish head first because the spines are all coming out of their face, but they're still alive. So I mean, I guess I'm not sure really to speak to like the chemical side of that or how the physiology of some of the species are able to manage not die from that. But yeah.
It's very interesting though. Thank you so much.
Yeah. I was like thinking maybe perhaps they don't have like the receptors to like get that poison. That usually happens for other amphibians maybe that don't really absorb like certain toxins, so they don't like get affected.
That's what I thought too.
Yeah. Awesome. And what let's go for the next question. How is tagging an important tool in fish studies, and can you explain how is it done?
Sure. So basically, when biologists studying fishes, they want to know basically as much as possible about the life histories of those fish. So how long they live, how fast they grow, where they move to or where they migrate to, how many are in a particular area, or also how many are caught by fishers. So there's actually a lot of different methods of gaining that information and learning that information about particular fish species or groups of species. But one of the more common ones is fish tagging or marking, or the combination of two. And so basically what it consists of the capture the tagging or marking of the fish with like a unique identifiable tag. So something with a number on it or some way of that you can catch the fish and know, okay, this one was already caught before, and then you release it. And then later on at a specific time afterwards, once that fish is either recaptured, resighted, or detected again, then what happens is you're able to get some of that life history information from those species or about that fish or that group of fish. And so what I mean by tagging, so there's a lot of different ways to mark or tag fish to help with these types of studies. And they really vary in the methodology and in the types of tags too. It can be something as simple as just catching the fish, cutting a piece of its fin off in a way that you know once you recapture that fish, you'll be able to notice that. You'll be, okay, that one is missing this specific part of its fin. This one was caught before, you record that information. They can be, you can have plastic Floyd tags or spaghetti tags that basically are inserted into the musculature of the fish just under the skin and stick outside of the fish's skin, and it'll be a bright fluorescent, piece of green or blue or orange spaghetti with a specific number on it. You can have some tags that are surgically implanted into the fish, like acoustic tags, where it requires either capturing the fish and actually performing a surgery on them, cutting them open, inserting the tag, sewing them back up, and then releasing them. And then also there are some that get really high tech and really expensive, like spot tags, the smart position and temperature tags, or PSAT tags, pop-up satellite tags, which actually can give you like long-term, exact movements of the fish, where it went, and the tag will be attached to the fish for a certain certain period of time. And then it can eventually, either when the fish surfaces, or it can be programmed to pop off at a certain time, goes to the surface and offloads all that information to a satellite, and then you can download that later on. And that gives you, long-term, like if you've seen, is it O-Search where they do the shark tagging in the Atlantic? That's primarily what they use, and you can see those tracks of the fish where it moved up and down the coast of the US or into the Caribbean or things like that. So, it really varies in terms of the methodology that you use and the types of tags that you use, but really what it depends on and the methodology or the type of tag that you use is the biology of the fish, the environment that it lives in, and also the type of information that you're trying to obtain from that study. So the types of questions you're trying to answer about those fish, and that really helps you narrow down which is going to be the most appropriate way to obtain some of that life history information that you're trying to gather about that species or group of species. So like for instance, some of the tagging that we've done here, they range from doing simple external garment tags or T tags, where you have a little gun, you capture a lionfish, and you basically just poke that gun into the musculature, the dorsal musculature up top in the back of the fish, and it just keeps a little green tag in it, and you release it, and then you go and dive and try to sight that fish again, or wait till somebody catches it and returns the tag to you. Some other ways we've done them is with more external tags, with the spaghetti tags, where you capture the fish, bring it onto the boat or bring it onto land, like for instance, some nurse sharks. We bring them into shallower water. And then you basically mark them in the dorsal fin, or in the case of some juvenile Nassau Grouper, right behind the pelvic fin, you insert the tag right into that area and then release them. And then you just wait for a certain amount of time for a recapture, resighting event, or for a fisher to return the tag to you and tell you the location of where it was, which would give you, movement information from where it was tagged and then where it was captured later on. And then some other tagging that we've done are some acoustic tagging with acoustic telemetry. So those are the tags that I was describing before, where they're surgically implanted into the fish. And the tags, once you release the fish, stay in the fish for the rest of its life. It doesn't harm the fish whatsoever. And for a certain specified period of time, that tag sends out like an acoustic ping, like a high frequency, just sound ping that if it is within range of a receiver, basically an underwater microphone, then that microphone then will record the information of that basically tag ping. So basically the ping gives out a code, and it gives out a date. And also you have the location of the receiver, that the where you actually deployed the receiver that listens for those tags. So you have the location of the fish too. And it basically gives you presence absence information. And from that presence absence information, then you can make inferences on movements and life history characteristics. So.
Nice. From at least the tagging methods I've heard, like they seem pretty intrusive. Is there any studies like studying like how much this affects their behavior later on in their life?
Yeah. So there actually has been a lot of work that has gone into that, because one of like the biggest assumptions and one of the most important assumptions that you have when conducting a tagging experiment is once you capture and tag that fish and then release it, that fish has to be one, unharmed, but two, act just like an untagged individual. You don't want the tag to alter its life history or its daily life in any way. Otherwise, you're not going to get the correct information. So because that is such an important part of every tagging study, there has been a lot of work that has gone into seeing like what are some of the best ways to complete fish tagging without really harming the individual, so that they will give you the most highest quality information that you have. And like, for instance, like one of the projects that we've been working on for a while is we were tagging fish at a spawning aggregation. And one of the hardest things when you're tagging fish or fish that exist or live in deeper depths, is when you pull them up to the surface, there's a lot of barotrauma involved. So there's a lot of overexpansion and pressure-induced stress, because the fish is at a deeper depth. When you bring it up to the surface, it has a gas bladder that helps it control its buoyancy underwater. Well, you bring it up so fast, the gas bladder expands and can really harm the fish. So a way that we alleviated that trauma and that stress to the fish is we started doing the tagging completely in situ, or in the water. So the fish that we were tagging were at around 40 to 50 meters depth, so between 130 and 160 feet. And we would trap, so we would capture them, tag them, and release them all at the depth of capture. So we completely alleviated that source of stress, and that would really ensure us that the fish are going to survive. They're going to be as least or less stressed as possible. And then once they are released, they're going to act like an untagged individual and, give very good data, and they're not going to go die, 20 days, 30 days later.
And on that project, you did 30 tags, and all of them were successful. Like, you never reported any losses on that, I believe.
Right. So yeah, for the most part, there wasn't any like tagging induced mortality or loss or anything like that from that study. They all, we all got a decent amount of tracking information after the tagging, so we could say, okay, if the fish did, if we suspected that there was tag loss or mortality later, it wasn't from the actual tagging itself.
And if you want to see that, we have like a video of that process on our Vimeo page. So I think it's like vimeo.com and then /Isla Mar EXP. And we have a short version and a long version. So if you are really interested in that, then watch the long version. If you just kind of want to see what it's about, then there's a shorter version too. And it just shows how essentially how Evan and the team did that tagging underwater.
Sounds really cool. Do you have any questions, Amanda?
No, I don't know if the question in the chat was answered already.
Yeah, it was about the grouper. Yeah. What are the juvenile Nassau Grouper preferred habitat type depth for your survey? Are you tagging the Nassau, and how frequently are you recapturing them? Are you trying to find the Nassau, um, I don't know, spawning aggregation with eDNA? That's a question.
Okay. Do you want to take the first part?
Yeah. So I'll take the first part. So for the juvenile Nassau preferred habitat, basically the habitat type we're finding is low to high relief hard bottom. Let's see. So that's the type. The depth we're seeing anywhere from 2 meters to about 10 meters depth is where we're finding the juveniles. So the low to high relief hard bottom habitat between 2 and 10 meters that is in close proximity to thalassia beds is what we're seeing thus far for our surveys that we're doing for juvenile Nassau grouper. And then yes, we are tagging the juveniles that we're able to capture. And we're still in the tagging phase of that. So we haven't had any recaptures yet. So we haven't had any recaptures yet. And then the eDNA.
Well, so we are with our eDNA project, and I guess we might touch on that a little bit later in the talk, but the intent of the eDNA project is in our first year is to kind of ground truth the process to see if we can locate a spawning aggregation using environmental DNA. And if we can show that is the case, then in our second year, then we're going to be going to suspected spawning aggregation sites and seeing if we can detect the presence of some of these fish, with Nassau being one of the targets at these like suspected sites or the unknown sites.
Super nice. So eDNA has not been used for this grouper type?
It has not yet. We are
okay.
we're the first project
Right.
to our knowledge that is using environmental DNA sampling to locate fish spawning aggregations.
Well, on to the next question. What can you tell us about your work in Tres Palmas, Rincon, and what makes it such an iconic landmark?
Yeah, so we've done a few things in Tres Palmas. Most recently, we helped HDR Reefscaping with the coral reef restoration work there following Maria and following the huge swell event that happened there in March after Maria. And so with that project, we helped basically transplant broken elkhorn coral that was already there on the reef. So we didn't bring anything new into the reef, but we just used the broken fragments and we cemented them back down to the substrate. And also some of the boulder corals that were toppled as well, we were able to right those and resubmit them to the substrate too. I believe that over that entire project, which we started in Tres Palmas, I think in like December of 2017, and we finished it in June of 2018, and we planted about 1,000 fragments of coral there in Tres Palmas. And we tagged about 10% of those. So like it's what an important part of doing a restoration project is to be able to monitor the success of that afterwards. And so we've actually put some tags, which are actually the yellow cattle ear tags that you'll see like on livestock. We've, in science and especially using things in the ocean, you got to get creative.
I've been there and I've seen them and I've been like, what is that?
Like,
good.
Yeah.
Wow.
They're not trash. Please don't.
They're not. I actually touched it to see what it was, but it's really in there. Like it's really in there.
Yeah. Yeah, exactly. So they're really in there. But actually in about a couple weeks they get grown over by algae, so we have to remember where they're at, but we've got GPS coordinates for it. And actually once you get used to seeing that shape, then you can pick it up pretty easily. But yeah, so that allows us to be able to go back to actually monitor the success of that. And we're working on a project proposal now to spend a little bit more time actually doing some of that post restoration monitoring. But besides that, we have always used Tres Palmas as kind of our backyard for teaching our field courses. So anytime that we have a group of students, Tres Palmas is essentially where we teach them how to run belt transects, how to do underwater visual census, how to learn
fish.
how to identify fish, how to do photo quadrats. And so we've kind of have this several years of data that we've also been collecting for the reserve through teaching these different field courses. So that's something that we've been able to provide to other projects to sort of look to see if we can observe any changes over time within the reserve itself. So it's actually part of the reason why I think it's such a beneficial place, not just for what the reserve is actually doing, but just the fact that it's close to shore helps with the education aspect of being able to show like our community what they have in their backyard. Whereas like Desecheo and Mona are also marine reserves, but they're so far and can be really inaccessible to probably the majority of people that live here. But with Tres Palmas, it's just right there. It's very easy for you to get in and snorkel and immediately see like the elkhorn coral and all of the fish, the parrotfish that are all swimming around the reef. And so I think it's a critical habitat in the biological sense, but then also for us too in the outreach and like the community like education side of it. It really gives us that opportunity to show like our residents, our students, our visitors, like why the ocean is so important and like what they can do to actually help conserve it.
Yeah, especially because it's so close. Would you say that it's more susceptible to like anthropogenic effects, like
Yeah, like I
contamination,
feel like
pollution?
the corals are so like high up, you know, people swimming with their fins could hit them very easily. I've seen that happen, so it's very susceptible to that too.
Mm-hmm.
Yeah, it is. I mean, and there's some river outputs that are kind of north and south of the reserve, and then when a really heavy rain, there's one area that will open up and kind of flood right out at Steps Beach too. So luckily, since there hasn't been as much coastal development right there immediately behind the reserve, it has alleviated some of that pressure. However, there has been coastal development again, like north and south of it, and so with the right currents, that can bring some of that sedimentation up onto the reef, and yes, that is impactful.
But that being said, you know, that is a definite concern. But one of the things that, you know, came out of some of the island-wide surveys and studies of the coral reefs, you know, around Puerto Rico, is that Tres Palmas does have some, you know, is one of the areas with the highest percentage of coral cover, you know, on the island. So yes, it is very delicate, you know, and it is very susceptible to some of those anthropogenic stressors and sources of land-based pollution. So that does need to be like very closely monitored. But, you know, as of now, you know, comparatively speaking, you know, it's doing fairly well. And I mean, you can tell as soon as you jump in the water, it's such a beautiful place. And, you know, I think it's just really like iconic of the island and
the Caribbean,
its beauty
overall.
in the Caribbean.
Yeah.
Yeah, that's amazing. That's amazing to see that it's so well taken care of too.
Yeah,
Like,
I mean, and you know, and that needs to be considered too, because you know, in the future, that should be considered in, you know, in monitoring development in that area, and you know, trying to keep, you know, the area as close to it is now, or at least, you know, limit the development that's going on in the area as much as possible, because it is such a, you know, a fragile and delicate and you know, very important, you know, reserve.
so besides that, what other projects is Isla Mar working on?
Well, okay, so part of being independent biologist is that you hustle, right? You're always hustling for projects. You're always writing new grants. You kind of have to be thinking a year ahead, if not more than a year ahead of what's going to be coming up. So we're constantly writing proposals for new work, monitoring to see like what the federal government and the state government is looking to, or what their interests are. And seeing like how our expertise can help answer some of those questions. So like, yeah, we're working on a couple things. I know this has been brought up, I think, already in our conversation, but we have a project right now with the Caribbean Fisheries Management Council where we're investigating like the juvenile habitat of Nassau Grouper, which Evan was talking about in terms of the tagging component of that. And we kind of answered some questions about that. So that's one that's one thing that we're hoping that we'll actually be able to expand on a little bit as well once we can kind of finalize the results of that and see what we've got from it and how we can expand on it. But right now that's on the east. It would be pretty cool if we could do a similar project to that over like along the west or in some of these habitats that we have in this area too that are similar to what we're finding there in the Fajardo and Ceiba area. We're also we're working on like our eDNA project. I think we could talk about that maybe a little bit more. For those of you that don't know, eDNA stands for environmental DNA, and it's essentially like the DNA that you can extract from an environmental sample. And an environmental sample can be water, soil, it can even be air. So what we are trying to do is visit sites out in the ocean where spawning aggregations exist, and then again, as I said, once we finish kind of our proof of concept, we'll be visiting some sites that are suspected to host spawning aggregations, and we're collecting water samples from these sites. We bring those water samples back to the lab, and we're doing this in collaboration with the University of Puerto Rico Mayagüez. So we're working with Dr. Nick Schizas in his laboratory, and we have a student helping us there that this is going to be part of her thesis. And so she essentially will filter all of those samples and then extract the DNA from those filters. And then from there is when we start our amplification step, where we start targeting like a specific gene that's found within these fish. And we try to see if we can actually detect their presence within that DNA that we can pull off the filter. Now, keep in mind, there's going to be a lot of other DNA that is on that filter, not just our target species. And actually, I like to phrase this as though it's like looking for a needle in a haystack, because that's literally what this is looking for just one particular species among DNA of so many other things in that system. But since we are focusing on spawning aggregations, then in theory, the target organism that we're trying to identify should be the most abundant thing at least in terms of the fish, should be the most abundant thing in the area where we're sampling. And so that's kind of how we based our rationale. And so right now with that project, we're finishing up, or we're working on analyzing like our year one samples, which was the sample collecting that we did at the known aggregation sites to kind of ground truth the actual methodology. And then once we have some results from that, we are going to then start visiting some of these suspected sites. So sites that fishers have kind of hinted to us like, oh, historically I used to see black grouper or Nassau grouper or Cubera snapper at this particular site. And so what we're going to essentially do then with the methodology is try to visit some of these places to see if we can detect the presence of these fish using eDNA.
Other projects?
Yeah, I mean, we can keep going. We got
so
we juggle a lot of things at one time.
Another project that we have in coordination and in collaboration. Sorry, that's the dog. in collaboration with HDR Reefscaping is nurse shark aggregation monitoring project around Puerto Rico. So same thing, looking at fish spawning aggregations, but specifically just with nurse sharks. There were some reported nurse shark aggregations around the island. So what we're trying to do is go do some monitoring work using drones to, quantify or count the number of individuals that are there. Also do some genetic work, looking at some of the population connectivity around the island, but then also looking at incorporating some tagging, which I described earlier. So some tagging to get some information, some life history information in terms of just population size or movement patterns or things like that. And we also have plans to use the BRUVs as well, similar as with the global fin print project around the island, looking at the shark populations, but we'll use the BRUVs to try to recite those tagged individuals later on.
Basically monitoring as they approach their aggregation site and as they leave their aggregation site to kind of look at timing for that. There's one particular aggregation that we've been monitoring now for several years with HDR Reefscaping. And this project that we're discussing is actually funded by the Department of Natural and Environmental Resources because they had an interest in understanding how effective the ban on harvesting nurse sharks has been, because nurse sharks are actually a species here on the island that's only catch and release. So you're not supposed to be consuming nurse sharks. And so another component of this project is we have a student at UPR Mayaguez, and this is again like her thesis, where she's actually she's visiting different fish markets, restaurants, talking directly with fishers and taking tissue samples of some of the shark meat that she has or that they have, and we're looking to see like what sharks essentially are caught around Puerto Rico. Mm-hmm. And I mean, we're hoping that we don't detect nurse sharks in those samples, but it's kind of a way for us to kind of like forensically, I guess, look and investigate to see if we notice that.
Yeah, and has there been occurrences where people actually like sell them? Like
yeah, there has been. Sometimes I think that it's that people don't know. So, but yeah, there have been some instances where we have heard of reports of nurse shark meat being sold, and that we've had come up in some of our samples too.
Right. So there's a question here. Are you working with the huge nurse shark in Hobos Bay? Fishers are from the area are complaining about pups following them and snapping at their ankles.
There is a component. That's not the one of the main main aggregations that we'll be visiting like frequently. But yes, that we are going there and at least trying to get some information from that aggregation, seeing when it happens, when it occurs, what time of the year, that type of.
But I hadn't heard about fishers saying that they're being followed by the pups. That's
interesting. Yeah. But yeah, we're aware of that, and one of the components of the project is to just survey the island, looking for different nurse shark aggregations that might not be known about. So the Hobos one, we know it's known about. I believe Hobonair has been monitoring it for a while. And then, but yeah, we have visited, or we will be visiting that one just to try to get, some information as well, information about that aggregation as well.
And could you guys, like, for our non-scientific public, could you guys like briefly explain what is spawning aggregation?
Mm-hmm. So a spawning aggregation is basically, it's a group of individuals of the same species that come together at a specific place and time for the reproduction or for the purpose of reproduction. All right. So in general, it has to be it's considered the general population size needs to be about 4 times that of what's normally occurring on the reef. And they're getting together for the sole purpose of reproducing. And it basically results in a mass point source of larvae into the environment. So fish, the majority of fish reproduce, and fertilization is external. So meaning they release their eggs and sperm, the gametes, into the water column, and those gametes mix, fertilization occurs, and then those fertilized eggs and drift at the surface of the water for a period of time, and then they develop into larvae. And then eventually those larvae further develop into kind of like little free swimming larval fish, which actually look nothing like adults. Like, you can be very good at adult fish identification, but somebody who doesn't know that much about fish larvae, you would have no idea. If you knew everything about, say, black grouper and then saw a black grouper larvae, it would look completely different. So those larval fish, then they drift in the water column until they find the appropriate habitat to settle, which is normally like near coastal environments, mangroves, seagrass beds, like nursery habitats. And then they dive down to the bottom, and then they start to grow to maturity. And then eventually, as they grow, migrate to deeper and deeper reefs, to where we normally are used to seeing them. So like spawning aggregations basically kickstart that whole process. It's more or less considered for that species like the main mode of reproduction, or at least the main like reproductive effort of that population. So they're extremely important for the persistence of fish populations, you know, at least the fish that utilize that type of reproductive strategy. Not all fish do, but some reef fish do. So they're very important to monitor and protect. And one of the downsides of having that type of reproductive strategy, like it's an amazing thing to see in the water. Like if you can imagine, some of the fish spawning aggregations around the world consist of, you know, thousands of individuals. So you can dive in an area and see 6,000 snapper, you know, in one area of the reef. You know, it's a biological phenomenon, and they occur basically, you know, usually around the same area, like at the same location and the same time each year. So it's really, that helps with some of the monitoring. But what we've been finding is that they were also heavily targeted by fishers over the past, you know, 10, 20, 30, 50, 100 years. Which makes sense. I mean, if you're a fisher and you're sustaining your family, your livelihood, you're making a living, it makes sense, you know, to go and fish during that time. You want to go when the fishing is good, you know, it's your best chance. So, you know, it's basic business, you know. But what we found, and what the research is telling us, is that these aggregations are very susceptible to overfishing. So that heavy fishing pressures during their reproductive time of the year actually has a hugely negative effect on their population. And a lot of species they're finding throughout the world that undergo this type of reproductive strategy are overfished. So much so that some of the aggregations no longer form. So we've talked a lot about Nassau Grouper. That's kind of a big, you know, hot topic right now with fish spawning aggregation research. And that's because if you just look at the actual like history, like the regional history of the Nassau Grouper, not just in Puerto Rico, but the, you know, entire Caribbean and South Florida and things like that, is Nassau Grouper for the longest time, they were the most commercially important like reef fish, fin fish species. So they were, you know, they had the highest amount of money in the fishery. That, you know, they were a lot of catch. Fishermen, you know, profited a lot off of fishing Nassau Grouper. That fishing was largely targeted to the spawning aggregations, and then they found that the aggregations were slowly becoming less and less and less, and fishing to the point where some of them collapsed and the population in the region began to collapse as well. And this went along for decades, and for so long that, you know, in 2016, the Nassau Grouper was listed as an endangered, or listed as threatened on the Endangered Species Act. So basically what you're looking at, you know, for the whole timeline, is you have a fish that was the most economically important fin fish species in the region, and then went to the point where it's now listed as an endangered species. So, you know, that's why, you know, it's just very important to continue, you know, these conservation efforts to preserve, you know, the fish spawning aggregations and this reproductive strategy. And you know, eventually, you know, the goal is to, you know, not just completely stop fishing for these species. You know, with some of them, that's the last ditch effort to, you know, save the species. But eventually, you know, if we're able to preserve and conserve the spawning aggregations, they'll continue to grow and grow and grow in numbers, so much so that eventually they'll be able to be reopened and be fished. You know, the populations will be able to sustain some sort of fishing pressure, so that, you know, the fishers in the region can begin to profit off of them again.
Yeah. What efforts are being done to try to communicate this with the fishers and try to, like, I don't know, like come to a happy medium, because it's also a pretty sensitive topic, because their economic feed comes from that source. So how is that outreach or communication done?
So there's a lot of different modes, I guess, that's been going on. I would say in the past 10 years or so, we've seen, you know, more of an effort to try to make like a education and outreach component and kind of shift the mentality from looking at spawning aggregations from like more of like a source of revenue or a source of economic benefit to really just like treating them more as like what they are, the biological phenomenon that's really just an important part of the life cycle that needs to be preserved in order for the persistence of the species. There's been a lot of different groups, you know, in the region that have a lot of like really good outreach programs. Like the first one, and I say this because this is basically one of, you know, the biggest grouper aggregation in the Caribbean is the one, the Nassau Grouper in the Cayman Islands, and the Grouper Moon Project, and they've done a really good job. They have, you know, live video feeds when they're on the boat doing the work at the aggregation seasons. They have, you know, the videos that they have with the Guy Harvey Foundation, and they've really done like a really, you know, good job of preserving that aggregation in the Cayman Islands, but then, and using their research to help like drive the management decisions to preserve those aggregations. But they've also done a really good job at getting that information out to the public, not just in the Cayman Islands, but also around the world, to kind of really drive that home and drive home the importance of this type of reproductive strategy. You know, like the Caribbean Fisheries Management Council now, they like in the past couple of years, they've been doing a lot of outreach and social media campaigns to really get that information out there as well, and drive home the importance of, you know, sustainable fishing and conservation of not just aggregations, but, you know, the fish and the island, you know, the fisheries island resources as well. So, you know, there is a lot of information out there on that, you know, regionally, but then also locally as well.
Nice. Is there any more questions, Amanda?
Yes, we have one question in this chat, and then we have another one in Facebook. So the first one here on the chat is, on average, how big, like, cover area is a spawning aggregation site for groupers, for example?
Groupers. So that depends. It varies because even though they all, the groupers will utilize this, what is called like a, you know, group spawning, or, you know, the spawning, they form spawning aggregations. Different species have different modes within that type of reproductive strategy that they actually use for reproduction. And each mode has its own like spatial component to it. And that also varies a lot based on the size or the number of individuals that are participating in the aggregation as well. So for instance, you know, say for, you know, just keeping Nassau Grouper, again, just using them as an example. A Nassau Grouper with 500 species, or I'm sorry, 500 individuals at an aggregation site is going to take up much less area than, say, an aggregation with 6,000 groupers, you know, just because the more groupers, the more space that they're going to use. And it varies, you know, Nassau Grouper are group spawners, you know, they form mass groups, you know, that with specific courtship behaviors. They gather, you know, in and around the aggregation site. Whereas like other grouper species form leks, meaning they come in earlier to the aggregation site, and the males build, you know, they designate their certain territories, they defend those from other males, you know, so they have a certain area that each individual occupies, so they spread out, you know, over the aggregation site, and then the females come in later. So that's a little bit different strategy than you know, the Nassau Grouper, which come together in just big groups and aggregate together. So, you know, there's really not an easy, you know, answer to that question, you know, because it's just highly variable, just based on the site, the number of individuals, and the actual species that we're discussing. but it can be, you know, a couple hundred meters squared to, you know, kilometers.
Wow.
And so, what would you say is the best way to track down spawning aggregations?
Like to locate them?
Yeah, I mean, yeah, it's a question from Facebook, so yeah, I think that's what he means.
Who asked
Yeah, that's hard too. Like locating them. I mean, you don't usually just blindly go after that. It's usually based on some kind of like traditional ecological knowledge. Like usually, like a fisher, somebody will kind of tip you off, I guess, in a way. I guess would that be like how you'd say it all starts really?
Yeah, I mean, one of the best ways and that, you know, researchers have found to locate these aggregations is using and building up relationship with fishers, because they are out on the water every single day. You know, they are out there, they're doing this for a living. They know, you know, where the fish are going to be, when the fish are going to be there based on temperature, current directions. Like they, you know, they really have like a wealth of just knowledge that they have gathered over the years just from that being their job, you know, and they're really experts at, you know, in some cases in finding fish. So, you know, that's building those relationships and input, fishers input, stakeholder input is extremely important in that type of research, because, you know, they've known about it for years. And, you know, I think a lot what helps is, you know, when you have fishers that have been doing it their whole lives and they start to notice that change, you know, okay, when I was, you know, 15, 20 years old doing this with my father, we used to catch this much, or we used to see this much, or we used to have this many different areas to fish. Well, now 40, 50 years later, I'm doing this and that's changed. We're not seeing as much, or we're fishing a different species. Or so the ones that, you know, have been doing this for a long time and have really noticed those changes and are willing to participate in some of the work and the data collection that's just an extremely important facet that, you know, is a lot of times is overlooked.
But there's other ways too. So like that's kind of like the starting point. But then it can be personnel intensive to try to locate a spawning aggregation, or you can take more passive approaches. Like one method that's used pretty frequently is like passive acoustics, which is like an underwater microphone that you can place down there. No divers have to be there really, except to actually deploy the instrument and then retrieve it. And so that underwater microphone then will just be listening to the soundscape essentially in this suspected spawning aggregation site, at least for individuals that are soniferous or that are actually like able to produce sounds. And so what we found with some of these fish will produce these courtship associated sounds. So sounds that are actually like specific to the activity that they are doing at the spawning aggregation site. And so those acoustic, hydrophones are able to pick up on those sounds, and groupers have really species specific calls or sounds that they make. And so with a really trained ear, you can pick up on what species of grouper, for example, is at that site. And then a good way then at that point to actually to verify it would then be to put divers in the water. So those hydrophones would be able to tell you like essentially like a range of when courtship sounds you start to hear like a lot more activity, and then you can put divers in the water there to verify that that's, you know, to get a count as well. When we go in the water, we're not just watching what's going on, but we are actually doing like a fish survey to get like a count of the species that are present, but then also to a size so that we can know something about the biomass.
And then divers too, on some of the like known or historical sites, you might know it was once a spawning aggregation site, but you don't know when it occurs or, you know, what it is. Sometimes divers will go in the water to try to find, you know, to see, you know, if you see an increased abundance of individuals that might show some interesting behavior that could be considered, you know, courtship associated behaviors, that would be a good indication as well. There's a lot of cool research being done with acoustics, as Chelsea mentioned, with passive acoustic monitoring, but then there's also some more like active acoustics using like echo sounders behind boats, sending, sound waves and pulses in the water. You can actually pick up, because as I mentioned before, like fish have swim bladders or gas-filled bladders that they use to monitor their buoyancy in the water. So when you're sending sound through the water, it hits that gas bladder of the fish and it ricochets off and bounces back, and you can actually detect a fish being in the water with those echo sounders. So you can basically drag or put one of those on a boat and zigzag or go up and down a specific track over a suspected site, and you can determine them that way as well. You can find aggregations that way. Hopefully we'll be able to give results of how you can use environmental eDNA to do that, but we'll be able to
more on that later.
Right? But that's a new and an upcoming technique as well. So there are a lot of variable methods to locate these aggregations. Even with that though, it is still very difficult.
Yeah, and sometimes you get lucky. You just happen to be in the right place at the right time.
Mm-hmm.
Yeah, and that seems like a field by itself, like all those acoustic stuff, like
yeah, it
like
is.
frequency analysis, maybe, and like trying to filter out like those frequencies. I don't know. Sounds pretty cool.
Still, yeah. Mm-hmm.
Could you quantify like biomass concentration with that?
That's the big question.
That's the big question. I think they are trying to do that with all or the abundance with acoustics, but they are really trying to do that with the echo sounders as well.
But right now, no. But that is kind of like that. But that's sort of the future of that specific field is trying to answer that question, because that's kind of the question that keeps coming up is, well, can you tell us how many fish are there by what's making the noise? And so we're still looking into that, but we don't have an answer to that yet.
Yeah, that'll be really useful for both research and like fishers.
Yeah.
Oh, that's really cool. Is there anything else, Amanda, in chats or anything?
Yeah, I'm just like internalizing everything that's happening right now, because it's a lot of information and it's amazing. Oh my God. Wow, I've never heard of this before. So thank you for sharing that. Mm-hmm. So to finish off, how can the general public basically be part of these projects and stuff that you have?
Yeah, so some of our projects required, like as you guys have heard since we've been speaking, like some pretty specific skill sets. And so there are some projects that we don't like take volunteers with us, but some projects in that sense might allow for like students or folks to come and like shadow what we're doing. So even if they're not able to actually participate in like the physical field work side of it, they might be able to just come along just to see like what it is that we're doing. So sort of like a day in the life of a field biologist. But then also too, we have some projects that do require volunteer help, especially our projects that we do at Tres Palmas. That's usually something where we'll ask folks to come help us conduct surveys, to help us go monitor the restoration sites. Those are skills that we can teach like pretty easily. And a lot of times it only requires snorkeling, so you don't even have to be dive certified for that. The best thing to do is to kind of monitor like our Facebook page, because that's where we will always post any like volunteer or internship opportunities that we have. And actually right before our interview today, I shared a link where there's like a form just to collect information from anybody that would be interested in volunteering, and that just sort of gives us like a basis, so like a foundation of folks that we can kind of search through if we see any particular projects that require certain skill sets, or that would be open to having folks come in and shadow. Yeah, I would say that those, and then occasionally too, we offer workshops that are just open to the community. Like last year we did a lionfish workshop that was open to anyone that wanted to learn about lionfish. And that was really cool. We did that in Rincon, and we had like over 60 people at that workshop, which was incredible. And we just we took some time just to explain like some of the misconceptions about the lionfish invasion and about eating lionfish. And then we also demonstrated to people the correct and proper way to spearfish lionfish and how to handle lionfish. So that's something that hopefully we'll be able to do again once everyone's able to meet in person. but yeah, so that's
Mhm.
those are some general ways. Like,
And I mean, we have to, you know, but besides the festivals, you know, once everything gets back to normal, we usually do like a beach cleanup or two, you know, every year at Tres Palmas. So we're always looking for volunteers, and you know, so yeah, putting out that usually is on our Facebook page, you know, different events, or you know, we collaborate with a lot of other organizations as well. That you know, we might just be a small part of a bigger, you know, festival or project. So you know, it's a good way just to, you know, we can put that information out on our Facebook page, and you know, we're looking for volunteers that way.
and then just kind of like specifically for students, like we really enjoy like enlightening students into this sort of this career that we've kind of established for ourselves in this way that you go independent, and it's kind of a non-traditional field, or it's a non-traditional approach to the field of marine science. So we also like to just, if there are ever any students that are interested in what else they can do in marine science, you know, besides being in academia or besides, you know, working for federal or state agencies, we like to kind of have that opportunity to have those one-on-one chats with students so we can explain kind of how we ended up where we are, because we really enjoy what we do. We think it's a very rewarding career, and so we're happy to be able to share about that experience with others, so that, you know, students getting into this field can see that there's a lot more to it, you know, besides what traditionally is projected as like the career path for a marine biologist.
Yeah, many of our followers are well students like in their undergrad. So what advice can you give them? Like what was the thing that like clicked to you when you were like an undergrad or like trying to apply for other graduate studies? Like what motivated you to do that?
Well, to touch on the first thing you said, like what advice would you give? I would say like I would say like take every opportunity that you have to learn a new skill, because you won't really marine science is a really diverse field and there's a lot you can do with it. And originally I thought that what I wanted to do was study specifically coral reef biology. And so I tried to get into doing undergrad research with a coral reef biologist, and it didn't really work out. I ended up approaching a fish biologist and got in to do undergraduate research with fish, and that was it. I was set. Like, fish is what I want to do. So I would just say, like, take every opportunity that comes up to be able to to test like a new skill or experience like a new aspect of marine science, because it'll really surprise you what you end up working with. And also to take as many diverse classes as you can in your undergraduate, because I will say that I do have one regret, and that is that I tried to avoid genetics and microbiology as much as I could in my undergraduate. And now I find myself working on a lot of molecular projects, which I had to go back and learn a lot about in my graduate studies. So I would certainly recommend to take like a diversity of things, because you really, I feel like when you get into graduate school, that's kind of where you start to solidify your interests. So if you can kind of diversify yourself while you're an undergrad, that'll really help once you get into graduate school.
Yeah, early on, like volunteer as much as possible. You know, look for different opportunities, even if it's something that you're not 100% sure you want to do. I would still, you know, welcome the opportunity, because you'll never know. One, you know, it's probably going to be a completely new experience, so you don't really know whether or not you're going to like it. But then two, you never know what that's going to lead to. You know, like not all. You know, like remember those memes, you know, from the, you know, that are always like, you know, marine biologists, what my parents think I do, what my friends think I do, and what I actually do. It's not all glamorous. It's not swimming with the dolphins, you know, all, you know, things like that. Like those opportunities might come about, you know, or if you want to go that direction, you can. But, you know, just always welcome new opportunities. You know, volunteer in the beginning as much as possible. Even if you don't like it, that's fine. Then, you know, you can, you know, do something else later on, but you never know what it's going to lead to. And, you know, it's not, you know, you'll get some bad opportunities. I mean, even now, sometimes, you know, you work on projects where you're like, man, I never in 1,000 years pictured I'd be doing something like this. And that can be both good and a bad thing too, you know, sometimes. So, you know, it's just, you know, definitely just diversify and take as many opportunities as you can.
That's excellent. Thank you for that advice. I think many of them will appreciate that, including myself. Yeah, anything else you want to add, Amanda?
I've actually met a lot of people, a lot of students that want to be marine biologists and they're like passionate about it, but they sometimes don't get enough like field work or enough like just experience in general, and they just get unmotivated. Like a lot of times it happens. So I think it's very important what you guys are doing, just like giving this experience to people and students like us, undergraduates. So thank you for that. We really appreciate it. Really, thank you. Right now, we can open the space to more questions. If anyone wants to ask any question, just they can open the mic and talk. We can give like 2 minutes for that, or just talk. We can just talk and converse. Is that how you say it? I don't know.
Yeah.
And I'll put our email in the chat here, and then if anybody has any questions afterwards, like they're welcome to email us. We're approachable people. We don't bite, and we couldn't do that from here. So
yeah.
No, but we're always happy to to listen and be able to provide any insight. I mean, one thing we really love about living here in Puerto Rico is that because it is a small island, and we get to know a lot of people, and the marine science community is amazing. And honestly, that has been what solidified our decision to stay here for good, is because the folks that we network and work with and collaborate with are just they're family, really. So that's we're happy to be able to expand our family.
Great. And no se escucha. Can you hear us?
Yeah.
I don't think Amanda can.
Oh, Amanda.
Yeah.
Oh, we lost her audio. Yeah.
Yeah, your audio. Amanda. Can you hear her? I can't hear her.
She can hear us, is what she's saying. I guess.
Mean, maybe if you turn off video, you might get better bandwidth for the.
Are there any other questions in the Facebook chat that you see? That maybe you can type?
No.
We can kind of hear
I can barely hear
but
you,
it's quiet. Yeah.
Oh my god, I don't understand. What the hell? Okay, we have one question in the Facebook chat. It was a long time ago, so it was about when you were talking about the research you did with the sharks. Natasha asked, what can be accomplished with the results?
Ooh, hopefully management decisions.
Oh my god.
No, but really that's kind of the ultimate goal. I would say most of the work we do has the goal in mind of providing either like data for things that are missing, so like knowledge gaps. So we're trying to fill knowledge gaps to be able to provide those results to the folks that are actually making the management decisions and enforcing that management. And so hopefully what we can find with, for instance, like the nurse shark aggregations, you know, since that's like a species that has a harvesting ban on it, like we're hoping to be able to show, or at least to identify where some of these aggregations exist, so that enforcement because again, it's during a really specific time of the year. So the idea would be that if there needs to be any like enhanced enforcement during certain times of the year, then we would be able to indicate like when and where that should occur. But then also too, we're curious kind of about the population of those nurse sharks. So we want to know like where they're coming from, where else do they travel around the island, and that's what some of the grub work with the tags is supposed to be able to help us with. And
how many are participating in the aggregations, if those are the same individuals showing up each year to the aggregation, and then also if those numbers are fluctuating, you know, over time, is another like really important, some really important questions as well to answer. And you know, and then that all can be used to develop, you know, if it's a data limited species, you know, whether you don't know much about it, you can develop somewhat of a baseline for that species, something to go off of. Okay, how is the population doing over time? You know, is it slowly getting less? Is it starting to grow? And then you can use that information to kind of as like an adaptive approach to then fine tune some of the management for, you know, for either that location or that species in general, to say, okay, what's going on right now may or may not be working. How can we make it better?
And that's essentially what the Nurse Shark project is going to be addressing, because there is really no baseline for that in Puerto Rico. So this project is going to provide us that baseline information that we can monitor thereafter. So for like subsequent years.
Okay. So you can basically do a lot of things with the results, basically.
Yeah.
Well, I don't see any more questions, so we could start closing. We at Amanda Sano, we wanna thank you both for this time. Like we really appreciate you took this time to be interviewed. We learned so much. I don't know about you, Amos, but I learned a lot.
Definitely.
And so yeah, honestly, we would all love to collaborate with you guys in the future with whatever project you have. You know, mostly, as you know, we're working with virtual stuff, but once this settles down, we could, you know, do cleanups and stuff, or just like us work with you and learn from you. Be like, please let me know whatever you want. So yeah, Amos, do you want to add something?
It was a really lovely experience. Honestly, I learned a lot as well. Yeah, and we're also like really available if you need anything, any help with stuff or promoting.
We have our emails and our Instagram, Facebook, and yeah, that.
Well, thank you guys. We appreciate it. This was great. I'm glad we had this chance to chat, and I'm thankful that you guys learned something, and I hope your other viewers, like on Facebook, that I know we can't see them, but hopefully they also learned something as well. But
yeah, thank you all very much. This is, you know, a great opportunity. We're very grateful and happy, you know, to do this with you guys, and then also have this interview, and you know, we're glad that you enjoyed it. And please, you know, like, yeah, if we'd love to have you guys, you know, if there's a project that, you know, we can use volunteers, if you're interested, we'd love to have you guys. And the same thing, if you ever need any help with anything, like, please don't hesitate to ask.
Yeah, like here at Amanda Sano, we have a group chat of a lot of like frustrated students that need experience.
Yeah, especially now with the pandemic.
Yeah, exactly. Yeah, so definitely like anything at all, just email me or anything. And so this video is gonna be on Facebook. You can also share it. You can like rewatch it over there too. And yeah, that will be all. That will be everything. Thank you so much. Thank you.
All right, thank you guys.
Thank you guys very much.
Thank you.
Have a nice night.
Yeah, thanks to all the viewers.
Bye.
Bye.
Bye.